In this video, we will discuss about some of the results that shows that interleukin 6 are upregulated in the cancer cachexia conditions. So, we have understood that cachexia is related to lack of motivations towards survival by the patients, and here they observe the results in the mice models. So, in this case, they use the three different kind of conditions: control conditions, pre-cachectic conditions, and cachectic conditions, and then do the principal component analysis of the plasma in the panel A, where they have find out that they can distinguish between control and pre-cachectic and cachectic conditions, but they cannot distinguish the cachectic with the pre-cachectic conditions from the plasma itself. So when they did it for the brain, they are able to see the three distinct populations uh, almost clearly, where they can distinguish control from the cachectic conditions and from the pre-cachectic conditions. And when they did the detailed analysis to find out which cytokines gets upregulated in both the conditions, they are able to see that the uh, interleukin 6 is interleukin 6 is one of the cytokines that get highly upregulated in the brain. And in the case of this, for the plasma also, we can see a distinguished change between the in the control and all the distinguished change in this interleukin 6 populations. For this PC2 loadings, indicating that for the control the interleukin 6 are lower, and for the case of this cachectic and pre-cachectic, they see interleukin levels are higher. And similarly for this PC1 loadings, PC1 loadings of the brain, they can see that these interleukin 6 levels are higher for this cachectic conditions. So thereafter they mainly focus on this effect of interleukin 6 on this cancer cachexia, as previous studies also shows a upregulations of the interleukin 6 cytokines in this cancer cachectic conditions. So they check this interleukin 6 levels at different conditions in the whole brain, and they are able to see that in the cachectic conditions there is a highly upregulation of this interleukin 6 compared to the control case and the other conditions that has been checked. And then in order to check from where these interleukin 6 are expressing, what they did is they parallelly inject this histec interleukin 6 and then check the interleukin 6 at different in the brains and at the different positions of the brain, and they are able to see that the tumors, they are able to see more interleukin 6 when they use the tumors. When they use this anti-interleukin 6, they can see a suppression in this interleukin 6. For the controls also, interleukin 6 are lower where they don't inject interleukin 6, and the control with anti-interleukin 6, they do not see any significant changes. So this shows that it's the tumor from where this interleukin 6 are being produced. Then they check it using another reagent, that is even blue, which is known for disruptions in the brain. Here also we can see that the even blue levels are higher in the case where the cachexia conditions exist, and when they use the cachexia conditions with anti-interleukin 6, they see a decrease in the even blue signals, indicating that it's the cachexia conditions that is causing the disruptions within the brain tumor cells. Then they check the other cytokines, that is IBA1, which is one of the chronic inflammations markers, we can say that, and it is upregulated. They checked it uh, in different conditions, that is in the control condition and the cachexia conditions, and at different sites. So in the NSE, they are able to see significant changes, and for the CTX and AMY also they are able to see the significant changes. But for the cytokines that is responsible for homeostasis within the brain, they do not see a significant changes for the NAC, HIP, CTX, VTA, MY, indicating that it's this NAC, CTX and AMY are being dysregulated in the Cachexia conditions, and they might somehow also plays a role towards these interleukin disruptions. Then they checked whether these suppressions of the interleukin using anti-interleukin 6 will have any impact on the survival rate or not, and they are able to find that when they use the tumors with anti-interleukin 6, they are able to increase the survival probability of the tumor cells, or survival life of the tumor cells, compared to the case where no anti-interleukin 6 are being injected. So this all results pointed out that it's the interleukin 6 that is somehow causing the cancer Cachexia conditions within the tumor cells, and also responsible for lower survival rate or less survival expectancy of the mice that are affected with the cancer cells, where Cachexia conditions are upregulated significantly higher, upregulated significantly higher in the conditions where interleukin 6 are being upregulated by the tumors. So that's all for this video. If you need to understand more about this results in detail, you can go to the reference that has been provided in the description. Thank you for your time.